Hello, I'm Minho Lee, a PhD student at Chungang University. In this video, I'd like to talk about the transport dynamics of water molecules, nanoconfined between lipid membranes. We studied the transport dynamics of water molecules confined between membranes to quantitatively investigate how the interaction between water molecules and membrane affects the dynamics of water molecules. We control the distance L between the membranes. We adjust this distance approximately from 50 angstrom to 80 angstroms. This is the system that I studied, and you might wonder, why do we need to study this system? The reason why we study this system is that such system exists in various forms within our bodies. They can be found in synapse, where neurons connect, the intermembrane space of mitochondria, where ATP is generated, and the endoplasmic reticulum, where essential proteins are synthesized. Here, I want to emphasize that molecular diffusion and diffusion control reaction occurring within this system play a crucial role in the vital activities of our life. That is why many researchers have been studying this system for decades. This work focuses on the diffusive properties of water molecules that influence molecular dynamics occurring within this system. In 2020, the research team from Keio University reported that as the distance between membranes decreases, the viscosity of the confined water molecule increases. They used microfluidics to show this result. However, the result was already predicted before. That prediction was made through the molecular dynamic simulation, MD simulation. 4 years before that paper was published, there were already at least 2 papers confirming that the viscosity increased as the distance between membranes decreased, based on the MD simulation. In those studies, the researchers analyzed the diffusion coefficients of the water molecules. They found that the distance between membrane decreased, the diffusion coefficients of water molecules also decrease. Since the diffusion coefficient is inversely related to the viscosity, this result essentially provides information about the viscosity. Therefore, we can consider MD simulation as simulation tool that can be used to predict experimental results. And from the simulation result, we can predict values that we haven't observed yet experimentally but expect to be measured in the future. Today, I'll talk about one of them that we hope to measure in the future. As I told you, we can determine the diffusion coefficient of water molecule confined between membranes. However, in this system, can the movements of water molecule be explained solely by a single diffusion coefficient? My answer is no, because the diffusion coefficient that I mentioned before is the average value of the actual diffusion coefficient. Here we can see the trajectory of the fewer water molecules that I analyzed. The water molecule close to the membrane is trapped and unable to move freely like the other water molecules. But the diffusion coefficient cannot give that kind of information. It just represents the average dynamic behavior of water molecules. In this case, How can we determine whether the dynamics of confined water can be explained by a single diffusion coefficient or not? It can be determined by calculating the non-Gaussian parameter, NGP. The NGP indicates how much the displacement distribution deviates from the Gaussian distribution. If the NGP value is not zero, it means that the movement of water molecule cannot be explained by a single diffusion coefficient. In case of bulk water, the NGP value is zero. This result indicates that the environmental surrounding the water molecule is highly homogeneous. In other words, the dynamics of water molecule can be described by a single diffusion coefficient. However, in case where the water is confined between the membrane, the NGP value is not zero for a long time. Over a long period of time, the NGP value eventually approaches zero. But sometimes it remains non-zero, particularly as the distance between membrane narrows. The NGP value starts from the higher value. The phenomenon of these NGPs had not been quantitatively explained, and my research gives a quantitative understanding of this phenomenon. To accurately describe the diffusive dynamics of water molecules, I consider two factors. First, I consider that the diffusion coefficient of water molecules varies depending on distance from the membranes, usually due to the interaction between head groups of lipid and water molecules. As the distance between membrane and water molecules, that is G is decreased, the diffusion coefficient decreased. And second, I consider that G can change over time. This change is caused by the diffusive dynamics of the water molecules. As the result, the diffusion coefficient of water molecule fluctuates. By considering these two factors, I derive the following equation, marked as yellow box. The first term on the right-hand side represents the diffusion of water molecules in the lateral direction to the membrane surface. And the second term on the right-hand side represents the transport dynamics of water molecules in the G direction. By using this equation, we can express the long-time behavior of NGP analytically. The key factor determining the long-time behavior of NGP is the fluctuation of the diffusion coefficient. I'll show you later that this equation can explain the NGP quite well. To calculate this value, first we need to know the environmental and dynamical properties of water molecules, including diffusion coefficients and density profiles along the Z-axis. So, we investigate various environmental and dynamical properties of water molecules along the G-axis. These results are consistent with the previous result. While varying the distance between the membranes, we confirm that there's a range of distance where properties of water molecule overlap. We define this range as interfacial water region. Regardless of L, properties of water molecules in interfacial region conserve. And beyond this region, the properties of water molecule recover their respective bulk limit values. These results are comparable to values obtained through different methods in 2018 and 2020. Using the profiles of water molecules, we calculate NGPs, which are shown as solid line in the graph. These results show a good agreement with the results obtained from MD simulation. From these results, we can say that non-zero NGP values comes from the fluctuation of the diffusion coefficient. Note here that when the distance L between the membrane decreased, the portion of the interfacial water increased, leading to an increase in the NGP value. We also calculate lateral displacement distribution in various times based on our equation. These results are accurately matched with the results obtained from simulation, represented as symbols. These results also show that our equation is correct. Here's the summary of my research. First, we explicitly show how the distance-dependent water dynamics gives rise to the non-Gaussian transport dynamics of the nanoconfined water. During that time, we also identify the interfacial water region near the membrane surface. Here, I'd like to thank all these people who helped me a lot. As a co-author and corresponding author, they gave me a lot of help. Thank you. If you have any comment and question, please feel free, let me know or send me an email. And please read my paper. Thank you.